Okay, today we're going to start on that part of the course that I kind of refer to as RNA weird stuff. We've been very DNA-centric so far, but now we're going to be learning a lot more about that other nucleic acid, which is very important and does some really interesting things. But I'm going to start all this with a reminder of things that you have heard or should remember, some of at least, from Cell and Molecular. So we're going to start with the basics and work our way to the weird stuff from there. So remember the central dogma. All right, don't laugh. DNA is used to create RNA, and the RNA is used to create protein. So DNA is replicated. The word is replication when you're talking about copying DNA to form more DNA. If you're going to form an RNA, you use the word transcription. If you're going to take RNA and form proteins from it, it's translation. So even at this stage in the game, people sometimes use the wrong word. Transcription versus translation can get confused. So try to get it right. So here's our diagram from our very old textbook that DNA replication occurs, transcription occurs, translation occurs to produce protein. So this slide diagrams a sort of important idea that genes are not transcribed equally relative to each other. Some are transcribed very heavily and produce lots of RNA, and some are transcribed very infrequently and produce very little. One thing that's not clear on this slide, though, is a lot of RNA doesn't guarantee a lot of protein, and vice versa. Okay, so here's our overview of gene expression in eukaryotes. We have our double-stranded DNA in the nucleus. Transcription by RNA polymerase begins at the promoter and then copies the DNA in a linear fashion without skipping anything. The introns are going to get removed, but they are part of the primary transcript, which is the first thing that's copied from one strand of the DNA. Following that, we have a series of processing events, including capping, splicing, and polyadenylation, which together prepare the pre-mRNA to form the mature mRNA that's going to go out to the cytosol by exporting it so that it can then be translated into protein. Protein translation mostly occurs in the cytosol. Okay, so transcription is in the nucleus and translation is in the cytosol. Don't forget that the exons are the part that are retained during splicing. Introns are the part that are removed. It's kind of a very badly used phrase, but the exon is the part that's kept. We also have to modify the RNA at both ends for stability, and also sometimes within the molecule. Okay, so to form the mature RNA, the pre-mRNA is spliced to remove introns, is polyadenylated, and has a cap placed on its 5' end. There are also other modifications that can occur, which we will discuss on some other day. Okay, so the binding of the protein, binding of RNA polymerase leads to transcription. Remember that the promoter itself and other upstream sequences are not transcribed. They are not part of the RNA. The RNA transcription start is after that. And transcription goes along the DNA without skipping anything. And of course, then we have processing. So here's a picture where it's sort of summarized, and which is very much like the earlier one. So we're going to talk about some of these processing steps, not necessarily in the correct order. Okay, so the cap is an unusual nucleotide at the 5' end of mRNAs. It includes an unusual base, 7-methylguanosine. It's got a 5' to 5' linkage, which is also very unusual. Importantly, the cap nucleotides are not encoded by the DNA. They're added shortly after transcription. And there can be other modified nucleotides that are present in the cap. Okay, so the purpose, one main purpose, is to bind the mRNA to the ribosomes for translation. It also protects the mRNA from degradation. Okay, so here's one version of the cap that you can see the first transcribed nucleotide is got a very unusual triphosphate linkage to what looks like an upside down nucleotide, which is our 7-methylguanosine cap. Okay, it's still got a ribose sugar, and the guanosine itself has been modified by the addition of a methyl group. So that's your traditional cap. Now, some versions of caps are actually a little more complicated than that. Your cap can also have, besides your 7-methylguanosine, it can have the first 1 or 2 sugars have a 2' O methyl group added as part of cap 1 and cap 2. And also oftentimes the first transcribed nucleotide is modified after transcription by the addition of a methyl group at the N6 position on the adenosine. So not all caps are the simple cap 0 type, with only the cap methyl being on the 7-methylguanosine. Other modifications of the cap are also possible. Okay, so here's what 2' O methylation looks like. Remember, the ribose sugar has an OH group at the 2 position, and many times the addition of a methyl group is used, partly to stabilize the molecule to protect it from nucleases. There are other examples of 2' O methylation that we'll see later. So remember, the 7-methylguanosine is a capping nucleotide, and you can have methyls on the ribose sugars and also M6A. Polyadenylation we will discuss next, even though it's at the 3' end. So eukaryotic mRNAs end with a non-templated group of adenosines, about 200 of them approximately. So non-templated means they're not present in the DNA sequence. They're not copied from a DNA sequence, whereas, of course, the rest of the mRNA is represented in the DNA. And importantly, like many things, polyadenylation has signals which direct the polyadenylation machinery to the correct location. Okay, for us, it's AAU-AAA occurs about between 11 and 30 nucleotides upstream of the place where the poly-A tail will be placed. Right, this is downstream of the translation stop codon, and it's within the 3' untranslated region of the mRNA. Now, it may be unexpected, but polyadenylation does not occur at the actual 3' end of the primary transcript. Instead, the primary transcript is cleaved or chopped at a location adjacent to that AAU-AA signaling sequence. So it's not the original 3' end of the primary transcript, which is one reason why we don't really know much about what eukaryotic transcription terminators look like, because that 3' cleavage product gets degraded very quickly, and so we don't get to see what it looks like. Another important thing about polyadenylation is that the enzyme that does it is a poly-A polymerase. It's not the regular RNA pol2 that transcribes protein coding genes. It's a different enzyme completely. RNA pol2 requires a template, whereas poly-A polymerase does not. So here's a picture of our polyadenylation signals, and where the DNA is represented, it's a double-stranded thing. Transcription gives you our pre-mRNA, and we're showing the location of the AAU-AAA signal for polyadenylation. Notice downstream of the actual cleavage site is the GU-rich region, and there's our 3' cleavage product, which goes away and gets degraded as part of the polyadenylation process. So after this endonuclease cleavage, we have the poly-A polymerase adding the adenosines to the 3' end in a non-templated way. I think I probably said all this. The steps are recognition of the signal sequence, cutting, and then addition of the non-templated A's, of course. So this is an oversimplified diagram, of course, because there are a lot of other proteins involved in the whole process, plus some other sequences that can contribute to it. Okay, so the last thing I want to go over briefly now is splicing, a process in which the introns are removed from the pre-mRNA. Exons are ligated together to form the final RNA that can then be translated in the cytosol. Splicing itself occurs in the nucleus. So there are signals for this, but much more is known about it than I will tell you. We will have a very brief overview now and have some more details on a different day. So the spliceosome itself is made up of proteins and small RNAs. The 5 small RNAs are called the uRNAs, and these uRNAs and proteins form complexes which are called snRNPs, which is small nuclear ribonucleoprotein particles, pronounced SNRPs. Each spliceosome consists of 5 SNRPs, and there's also a bunch of other proteins called splicing factors, which are involved in the initial stages and also in the regulation of the splicing process, something we'll discuss on a different day. So here's a picture of a spliceosome. Very diagrammatic. It never looks like this. But the idea is, of course, there are signals. And the ones that are generally understood by most people is that the first two nucleotides of the intron are generally GU, and the last two nucleotides of the intron are generally AG. Close to the 3' end of the intron is an important A, which is involved in formation of the lariat structure, which is part of the splicing process. So this is just to give you an idea, thinking about it, that some introns are very, very long. Some are like thousands and thousands of nucleotides long. And so there's got to be much more to the recognition than just your good old GUAG. Well, there is a lot more to the recognition. And some introns are spliced more efficiently than others. So this is just to give us an indication that there's a lot more that goes on here besides your basic GUAG thing. But this is true. The intron borders generally are those. They are spliced out with the intron, so you won't see them in the final transcript. And there's a particular A in the intron near the 3' end of it that will form the branch point or lariat as part of the splicing process. Here's our picture again. Okay, so now we've got everything done. We've made a mature RNA. We've capped it, polyadenylated it, and spliced it. And then it's ready to go out to the cytosol for translation. Now it's important to realize here, and sometimes people forget this, that your mature mRNA, it has its 5' cap, but then there's a portion of it which is before the start codon that's going to start translation. Now our start codon is going to be an AUG, and it's not at the very 5' end of the RNA, not by a long shot. There's always a region preceding it, which is the 5' UTR, 5' untranslated region, which is before the start codon. If you think about that, it makes a certain amount of sense, because a ribosome is a pretty big thing, and it needs someplace to land so it can slide along and use our start codon. Okay, following the 5' untranslated region, we have our start codon. Then we have our codons, which are, of course, read in groups of 3 to give our protein sequence according to the rules of the genetic code. At some point later, we have a stop codon in frame with the original AUG, and that defines the termination or end point of the protein. Following that, and people tend to forget this also, there is a region called the 3' untranslated region, and that is the part of the mRNA that follows the stop codon that is not part of the translation part. So the codons within the 3' UTR, not codons, nucleotides within the 3' UTR are not, they don't count as codons because they're not read in groups of 3, they're not translated, but they can serve and do serve some very important regulatory functions, some of which we'll see later. Okay, translation overview. And again, we're being very sketchy, schematic here. Okay, so now we've got our mRNA ready for translation. It has an ORF, or open reading frame, and that's where the codons are. That's what can be read to produce the protein. It starts with an AUG, ATG in the DNA, and it dictates the reading frame of the protein, the way the nucleotides will be read. There will be eventually an in-frame stop codon to end the formation of the protein. And then, of course, we have our 3' UTR and our poly-A tail. So translation overall, the process, it proceeds 5' to 3' on the mRNA. So it's moving near the beginning of the mRNA towards the 3' end. The first translated codon will be the end terminus of the new protein. So you will have a methionine codon at the end of, as the first amino acid in your protein. So translation moves towards the C terminus of the protein. And the key players in this, of course, besides the mRNA itself, are the ribosomes and the tRNAs. One thing that's kind of hard to visualize is what this looks like when the ribosome is actually translating the mRNA. And that's because there can be many, many ribosomes together associated with the same mRNA. It's not like it goes one at a time. So these are called polysomes when you have multiple ribosomes all together translating the same RNA at one time. Okay. And this is what is sort of conceptual, what it looks like, that you've got your 5' end, you've got your AUG, where translation will start. And so what this is showing is a bunch of ribosomes all moving along the same RNA in the 5' to 3' direction. Now the little green thing that's coming out, starting with the AUG, it gets longer and longer. That represents the protein that's being synthesized. So as the ribosomes are moving along, the protein gets longer and longer, and eventually when the stop codon is reached, it will get released as a new protein, and the ribosome can then fall apart and start the process all over again on a new mRNA. Now, importantly, the tRNAs, these match the correct amino acid to the correct triplet codon, and these are, of course, crucial for the translation process. There can be no more important function than that, because they're basically converting one form of molecular information, which is to say RNA, to another form entirely, which is the protein. So the actual matching is done by aminoacyl tRNA synthetases, which correctly attach the right amino acid to each of the correct tRNAs matching that codon. So here's a representation of what a tRNA looks like. Notice the extensive RNA-RNA base pairing here. It folds into this so-called cloverleaf structure, where they've got specific regions. There's the acceptor end, where the amino acid is attached, and then you have the anticodon loop, and that's the part that's going to match up with the mRNA. So this original primary transcript folds up to form this interesting cloverleaf structure. Notice also there's some unusual nucleotides here. We've got some that are designated Psi, which is an unusual form of uridine, and then we've got Ds in here, right? Those are dihydrouridines. And there could be many other modifications, many modifications to the tRNA molecules, not shown here. Okay, so they're amazing little molecules. They're only about between 70 and 100 nucleotides each. So the important things you need to know is which part is the anticodon that matches the tRNA to the mRNA by a form of base pairing, unusual form sometimes. And this ultimately matches the amino acid to the genetic code. The amino acids attached to the 3' end. And so really tRNAs are the adapters between the RNA sequence and the protein sequence, converting one type of information into another. Here's a representation from your old textbook of what this looks like. And I'm going to tell you in a sec, this exact base pairing they're showing is not universally true. Okay, one sometimes gets the impression you've got 61 sense codons. There are therefore 61 tRNAs, one for each sense codon. This is not true. It doesn't work that way in ways that I'll show you in a sec. But this shows your loaded charged tRNA in the process of participating in protein synthesis. Okay, here's our genetic code. Remember that the way the genetic code works, you've got some amino acids are encoded by numerous, as many as 6 different codons, whereas some amino acids, namely methionine, is coded by only one. So there are lots of nucleotides that are modified in different ways, especially in tRNAs, but also in ribosomal RNAs and in mRNAs. Okay, so these modifications include things like 5-methylcytosine, inosine, which we'll talk a lot about, 1-methyladenosine, pseudouridine, etc. And people are working hard to try to fully understand what the effects are of those modifications on the functions of tRNA especially, but also mRNA and others. Okay, so the inosine at the anticodon is one example of how some tRNAs can read more than one codon. So I said a second ago, there's not one tRNA for each exact codon, and a lot of the reason for that is some tRNAs can read more than one. So first of all, what is inosine? If we start off with adenosine, we can see it's got a big old amino group sticking off, that if that is removed by a process called deamination, that is replaced by a double bond O at that position. So that nucleoside is now inosine. It started being adenine, but by this deamination process, it's converted to a different molecule, which base pairs differently from the initial adenosine. Okay, so it's a modified form, and its occurrence within the anticodon loop is important in terms of its ability to cause that tRNA to read more than one codon. The inosine changes the base pairing properties. In mRNA, it pairs like a G rather than an A. In tRNA, it can actually base pair with more than one codon. So if it occurs in a codon, it can cause a change in amino acid sequence. If it occurs in a tRNA, it can allow reading of inexact codons. So remember, inosine, it allows, well, we'll see in a second how inosine and anticodon allows the same tRNA to read different codons. It usually occurs at the wobble position in the anticodon. Here's our genetic code again. Here's our tRNA again. Okay, so the strange codon-anticodon pairing. Remember, the genetic code is degenerate, which is to say many amino acids are encoded by more than one codon. For example, CCU, CCA, CCC, and CCG all encode proline. But that doesn't mean you need 4 tRNAs to encode proline. So remember, if you're inosine, you don't have an amino group. Instead, you have an O. And in tRNAs, it can base pair with cytosine, uridine, and adenosine. So tRNAs with inosine in their anticodon loop can therefore read, in some instances, 3 different codons because of this altered base pairing property of inosine. So here's a picture. An inosine can base pair with a cytosine. It can also base pair with a uridine. It can also base pair with adenosine. It takes a little contorting a little bit to get it to do that, but it does do that in the context of a tRNA anticodon loop. In mRNA, generally what you have is inosine base pairing with cytidine or cytosine, because it mostly behaves like a G. In the context of a tRNA, it's got more options. Okay, so here's an example of one glycial tRNA that will base pair with 3 different codons. For example, we've got glycine codons, we've got GGU, GGC, GGA, GGG, are all glycine codons. Well, it turns out that there's one tRNA which can actually read 3 of those. We have a CCI, which is inosine, which can read the GGU, GGC, and GGA, but not the GGG. Okay, so you should think that wobble pairing is always involving inosine. It doesn't. In the context of a tRNA, G can often pair with U, not just C. So it's not an example of a modification, it's just an altered form of base pairing that occurs in tRNAs. So for example, one phenylalanine tRNA can read both of the phenylalanine codons, which are UUU and UUC. So here's a picture of that. We've got a phenylalanine tRNA base paired with its actual base pair with a GC at the 3rd position in the codon, the 1st position in the anticodon. So that's a good old GC base pair, just like you would expect. What's not so expected is that we've got a guanosine, which can also base pair with a uridine at that position. So this UUU codon can be read actually by the GAA read 5' to 3' on the tRNA. So wobble pairing is not just a matter of inosines. There's other forms of base pairing that can allow tRNAs to read more than one codon as well. Okay, so there's no need to have 61 different ones. But the variants do encode the same amino acid, or else things would get very confusing. Okay, so now I'd like to talk a little bit about inosine as it occurs in mRNA and its effects. Okay, so the enzymes that are involved are called ADARs, which is to say adenosine deaminases acting on RNAs. These can deaminate adenosines with encoding regions of RNAs as well as elsewhere. So the occurrence of these I nucleotides changes the amino acid sequence of certain proteins. The I is read as a G by the translation system, and also by reverse transcriptase, and also by the splicing machinery. So you can alter many features of gene expression by specifically changing certain adenosines into I, which is read as G, by these enzymes. Now, adenosine deaminases also work on tRNAs but those aren't called ADAR, those are called ADAT. There are also adenosine deaminases that act as during purine degradation, and those are not ADAR either. So this is a specific class of adenosine deaminases that act specifically on RNA. Okay, again, here's adenosine, can be deaminated to form inosine, which alters its base pairing. So famous examples of, this is relatively rare in mammals, but it does occur. So famous examples of this editing include the serotonin receptors in brain, glutamate-sensitive calcium ion channels. This I'll say on another day that this is an example of what's considered site-specific editing versus the more promiscuous editing that occurs in 3' regions of the mRNA and in repeated sequences. Okay, oddly, some organisms, which is to say cephalopods like squid, use RNA editing very, very extensively, whereas for their protein coding regions, whereas mammals use it, but not nearly as frequently as squid. Okay, so here's two examples schematically of what this type of editing looks like. We'll talk about the double-stranded nature of the substrate next time, but notice the first one on the left. We have ADAR2, and the type of editing it's going to perform is called Q to R editing. So what's happened is a RNA sequence which base pairs to itself. There's a particular codon in exon 11 of this receptor, which is going to be edited. So what's going to happen is we've got a CAG codon there, which encodes glutamine. It is modified to form CIG, which is read as CGG, which is, if you look at the genetic code, an arginine. So by editing this particular codon to a CIG, read as CGG, we've converted that amino acid, and that actually does alter the function of that particular protein. The second example is more elaborate. There's two different active forms of these ADARs in mammals. And again, we've got an exon base paired to an adjacent intron. And this time we have 5 different editing sites, which can give you a whole bunch of different forms of edited RNAs. So in the first one, we start off with our genomic copy. The starting mRNA sequence is an AUA, which reads as isoleucine. Now that second A can be edited to form an I, read as G, so that will then read to the ribosomes of the tRNA as AUG, which is methionine. In the next one, we've got an AAU, asparagine, can be edited in 2 different ways to give you IAU, AIU, which are respectively read as aspartic and serine. So you can have double editing events as well. The last one is another isoleucine, AUU, is changed to IUU, which is read as GUU, which is valine. So this particular serotonin receptor can be modified in different ways to give a variety of different receptors, which have different properties from the original starting one that was encoded by the genome. It's slightly more representative without the double-stranded RNA, the types of editing that can occur at this particular molecule. Okay, again, here's our genetic code for some reason. Okay, so there's a worksheet we're going to be looking at, and I'm going to go over some parts of it, and then you're going to take it home and do it, and then turn it in next week. All right, so the first question on the worksheet, and I think I did this next time, we're going to say, for example, this is a little unusual, that the central A of codons of the CAN can be edited, and you're asked to go through the possibilities as to what the original codon coded and what the edited version. So you simply take the CAA, change it to CGA, etc., and read the genetic code to see what that says. And another question we've got similar, but this one's even easier. We have a methionine codon, which we know is ATG in the DNA, and then we've observed that we look in the cDNA, we see a valine instead. So the question is, what editing event would have given you that valine? So there's only one possibility. It used to be ATG. There's only one nucleotide that can be edited, which is the A. So we'll now read that as ITG, interpreted as GTG, which encodes valine. So we'll see the worksheet next time.